Okay, can you guys hear me?
Yes.
Yes.
Perfect. So I apologize. I forgot up my phone, and we have this whole Duo mobile link login thing. So anyway, that's why I'm late. So does anyone have any questions about anything right now? Okay, so we'll just pick up where we left off before. So we're talking about what goes in and what goes out. And so I'm just gonna summarize on this screen. So the first thing we have is glycolysis. I'm gonna move my screen around because it's hard for me to write this way. Okay. So we have glycolysis, and then remember what goes in is we have glucose, and then we have to put in 2 ATP. And then what comes out is, hang on one second. Screen sharing is paused. Wait. All right. Okay. So can you guys see the screen now? Sorry for the technical difficulties.
Yeah.
All right. So we have glycolysis. That's the first step. Glyco is sweet. Lysis is to break apart. The glucose molecules get broken down by your stomach and then go into your blood through your small intestines. And then we have 2 ATP that, remember, we have to put in. This is our energy investment. We have to pay to get energy out. And so in the end, we make 4 ATP. Hang on a second. Okay, so we have 4 ATP that are out, and then it cost us 2, but we've been made 4. So our net, we'll just keep it a track down here. Net is 2 ATP, and then the next step is that we at the end of, we also have pyruvate. We get 2 NADH and 2 pyruvate, and those pyruvates still have energy in them. So the step 2 is that we're going to take the pyruvate and we're going to move it into the mitochondria. So we'll just call it into the mitochondria. MT is short for mitochondria. And so what goes in is 2 pyruvate. And what comes out is 2 NADH and 2 acetyl-CoA. And we talked about carbon dioxide, but I really don't care if you know that or not. And then in step 3, we have the Krebs cycle, or the citric acid cycle. And in there we put in 2 acetyl-CoA, and we also put in to the Krebs. Well, that's it. We put in, and then what comes out is 2 ATP. These ATP we have to use for step 2 here. So we get 2 ATP out of the Krebs, and then we get 2 FADH2, which is the electron carrier. And then we also get 6 NADH, which we got here, and we also got here and here. So in the end, we have 10 NADH, 2 FADH2, and 2 ATP. And so anyway, and then we take these and take the electrons off of them and put them through the electron transport chain. That pumps protons across the membrane. So the whole goal is the electrons flow through the membrane. They attract the positively charged protons. And then we get this big buildup of protons on one side of the membrane, on this side. This is the outside and the intermembrane space. And then this is the matrix. And so we have a bunch of protons on one side of the membrane, and they, just like if you had a buildup of water or something, are going to want to flow back in. That's because of diffusion, which we talked about in chapter 8. So these things, these protons diffuse through this ATP synthase, causing this to rotate, and I'll show you how that does that. And that kind of works like a vacuum cleaner motor or an electrical motor. So this is the parts of it. F0 is in the membrane, so it's inside the inner membrane. Let me erase this real quick. Okay, so we have our mitochondria. It's a double membrane. So it's got the outer membrane and the inner membrane. And so we're talking about this space here. This inner membrane is phospholipid bilayer. And this is where the electron transport chain is. And then ATP synthase is sitting in this membrane too. So this is F0 in the membrane. And then F1 drops down into the inside, which remember we call the matrix. This is the inside part of the mitochondria. And so this is kind of flipped upside down, but you have this thing that rotates. This is what rotates, F0. And it, like a car's drive shaft, it turns this rod, which is this thing right here. You don't need to know though that it's gamma, but it's made out of protein. So this is a subunit. This is a gene that codes for RNA, which makes the protein that builds this thing. And then we have alpha subunits and beta subunits. And so all these subunits come together in sigma and epsilon. All these subunits come together to build this F1. So we're just going to call it F1. You don't need to know all this stuff. So what you do need to know is F1 is located in the matrix. F0 is located in the inner membrane, which it's upside down and backwards, but it's written right there. The protons go through here and make this thing rotate. And when it rotates, it opens and closes to build ATP. Okay. So I'm just looking for these videos.
Hello?
Yeah, I'm looking up a video. Okay. So I'm going to. Okay, so can you guys see this video?
It still shows the slide.
Okay, how about now?
Yeah.
Okay, so this is the F0, the thing that's rotating. You can see the green circles. Those are the hydrophilic heads, and then the yellow squiggly lines are the hydrophobic tails of the phospholipid bilayer. So this is the inner membrane of the mitochondria, and this thing that's spinning right here is the F0. So the way that this works is, remember in the electron transport chain, we pumped all those protons, which are represented by these blue circles here. So the protons are built up in the intermembrane space, and that means they tend to flow into the matrix, to the inside of the mitochondria. So what's happening here is that the proteins have those R groups that we talked about in chapter 5, and in this particular case, the red ones in the F0 protein are negatively charged, and remember the blue protons are positively charged. So they're attracted to each other. And when the blue ones attach to the red negative, now they're positively charged. So that fixed blue positive in the middle, it repels. So two blues repel away from each other, and then a red and a blue would be attracted to each other. And that's exactly how like a vacuum cleaner motor works, or an AC motor. By changing the attraction or repulsion, it only spins in one direction. And you can see this thing is actually spinning, in this case, clockwise, because of those protons going from a high concentration to low concentration. And that spinning motion creates force. And I'll show you what this looks like. Okay, so hang on. Okay, can you see this other animation?
Oh, we can.
Now can you?
Yes.
Okay.
Yeah.
So this is looking down. This is looking down at the bottom of that F0. And so in the middle, this thing right here is that sort of that drive shaft. And then these yellow things are the beta, and the red things are the alpha subunits. And then the green things are actually representing ADP. And so I know that this is like a complex molecular machine, but think about it this way. Let's say that I gave you some balls and sticks to build these molecules, and we know that you guys can build these from a chemistry model set. So let's say that I gave you a whole molecule of ATP, and I gave you a phosphate. How would you put it together? It's not a trick question.
Can you
Sorry,
repeat that?
Yeah.
Yeah, so if I give you a molecule of ADP, adenosine diphosphate, and a phosphate to make ATP, how would you make ATP?
You combined them? Or no?
Yeah, that's exactly right. You take one hand and have a phosphate in it, and your other hand and have ADP in it, and smush it together, right?
So it becomes di, becomes tri, right?
Right. So what this thing is doing is it's literally taking a phosphate and an ADP and smushing it together. You can see that the red things are actually moving back and forth, right? And they're literally squeezing a phosphate onto an ADP molecule, which is kind of cool, right? Because that's how you do it. And so that's how this whole thing works. These protons spin that F0, which is like a car engine or a vacuum cleaner motor. It makes force to turn this thing, and it's literally squeezing the ADP and the phosphates together to make ATP. And so this is just a recap of like what I just showed you. So we have the intermembrane space. Remember, so in chapter 6, you guys learned this about the mitochondria, but we have an outer membrane, an inner membrane. The space in between is called the intermembrane space, which is the space between the inner and outer membrane. And then in the inside is the matrix, and this is the inner membrane. So this is F0, right? That's this thing right here that I showed you spinning around. And these protons come in here and they're attracted to this red negative. And once they bind there, now there's two positives and it makes it spin. This is this piece right here. Right there is this, what we're looking at. And these subunits, this blue one is this red, and this white one is yellow. So it's like you're standing underneath it looking up. So this is the bottom of it. And this binds ADP, and this binds a phosphate, and it literally smashes it together to make ATP. And that's why it's called ATP synthesis, because that's what it does. It synthesizes ATP. This is just the same thing in what we're talking about. This is from your text, I believe, or maybe I took that out of a genetics text, but this is that thing, same thing we were looking at. So this is that drive shaft that you don't need to know that, but this is going on 3 sides simultaneously. And just to remind you, right, this thing has 1, 2, 3 alpha and betas. So that was what we're looking at. So in this configuration, when it's open, the artist is showing that it has ATP in it, and that's released. So it's getting rid of that. Once the ATP is gone, this thing rotates like we showed, and it changes its shape. So now it fits ADP. So the ADP binds into it. On this side, it's releasing ATP, right? So it's just, the artist is showing you that this stuff is going on each side simultaneously as this rotates in different steps. So ATP is the green, right? ATP is in green. Then it changes its shape. So now in the blue conformation, it fits ADP. And over here, this would fit ADP as well. This would also fit ADP. And then this rotates some more, and we have the phosphate that's getting smashed together. So that's what's going on here on this side at this point. That's what's going on here in the red one at this point, and so on. And then it rotates again. The ATP is created. It rotates again. It opens and frees it. And so while it's spinning, it's making ATP on 3 different sites of the same protein. Does that make sense?
Yes.
Okay, cool. So it's a multitasking. These dudes won a Nobel Prize for chemistry for figuring all this stuff out that I just taught you in 1997. So this is when we knew how our energy to stay alive came from, in 97. I would show you a video clip, but I'm not sure where this clip is. But basically, this is an actual electron microscope of this, and you'll see that when the membrane is intact, this thing spins. And when the membrane is punctured, it doesn't spin. And so we know that ATP synthase is what is making that ATP. All right, so it's kind of a cool system. I mean, if you think about it, your body's using electricity, which we just learned about from, you know, Ben Franklin a few hundred years ago, to spin a motor that's just like a vacuum cleaner motor that wasn't invented until the late 1800s, to make energy for us to live in the form of ATP. And we know that through glycolysis and all that other stuff, so glycolysis, and then we have the, you know, moving it across the membrane into the mitochondria, and then the Krebs cycle, and all that, we get 2 ATP. That's not enough to live, because that's what we make when we hold our breath. And from the electron transport chain, we get 34 ATP. And I'm just going to break it down how this works. So remember, in the electron transport chain, we have complex 1, and then 2 doesn't span the membrane. And then we have coenzyme Q and complex 3 and complex, well, then we have cytochrome C and then complex 4. So there's 3 complexes that NADH goes through. And remember, it has 2 electrons. So that means it's pumping 2 protons here, 2 protons here, and 2 protons here. So for every NADH, we pump 6 protons. 2 plus 2 plus 2 is 6. And we need, every 2 protons can make 1 ATP using ATP synthase. So if we break it down, every NADH pumps out 6 protons. We need 2 ATP, or 2 protons to make 1 ATP. So we just take 6 divided by 2, and that means that for every NADH, we can make 3 ATP. Remember, FADH2 doesn't go through all of that. It only goes through complex 3 and 4. So we only pump these 4 protons. So FADH2 pumps 4 protons out because it only goes through 3 and 4. Again, we need 2 protons per ATP. So 4 divided by 2 means we get 2 ATP for every FADH2. All right, so then if we just go do the math, we know that we get 10 NADH. Remember, we get 2 from glycolysis, 2 from making acetyl-CoA, and then 6 from the Krebs. So that gives us 10. We multiply that by 3, which is how many we get for each one, and that gives us 30 ATP total per glucose. And then we have 2 FADH2 from the Krebs cycle. We get 2 ATP for each one of those, so that's 4 ATP. So this all comes from the electron transport chain. That means we get 34 ATP from electron transport chain, and we get 2 ATP from the Krebs cycle. So we get a total of 36 ATP per glucose molecule to use for energy. Does that make sense to everyone?
So the electron transport chain gives us 36 ATP total?
No, 34. We get 2 from the Krebs cycle.
Oh, 1 second. 34. Okay.
So here's the Krebs. Sorry, I'm eating lunch. And we have this ATP that's being made from the Krebs cycle. Remember, there's 2 acetyl-CoAs, so we get 2 ATPs. Okay. So here's a quick question. Bacteria can make 38 ATP per glucose. Humans can make 36 ATP per glucose. Why are they different?
Hmm.
So we actually made 38 ATP, but we lost 2 somewhere. Do you remember where that happened?
Glycolysis?
Well, after glycolysis, we had to put the pyruvate, we had to move the pyruvate into the mitochondria. Remember, to make acetyl-CoA? And that cost us, 1 ATP per pyruvate. So it cost us 2 ATP. Now, why would bacteria get to keep those ATP?
They don't use as much energy.
No? Do bacteria have mitochondria?
No, I don't believe so.
Bacteria have no membrane-bound organelles, right?
Mhm.
So because they don't have to pay to put their pyruvate into the mitochondria, it's all made in their cytoplasm. They keep those 2 ATP that we have to spend to put in the mitochondria. So, but does that mean bacteria are more powerful than us?
No,
Why not?
because they don't have membrane-bound organelles. They're not as complex.
Well, the reason is remember, you can have lots of mitochondria in a single cell. You can have thousands of mitochondria in a muscle cell, which means that you're making 1,000 times more ATPs than bacteria make because they only have 1 cell. Does that make sense?
Mhm.
So we can actually make a lot more energy because all we have to do is make more mitochondria in the same cell. And that's what I want you guys to kind of get a grasp of, is that the amount of mitochondria vary between the amount of energy the cell needs. So your skin cell doesn't need a lot of energy, so it doesn't have a lot of mitochondria. Whereas your muscle cell needs a lot of energy, so it has a lot of mitochondria. And we can vary the number of mitochondria in every cell. All right. So let's play a little game here and just see if you guys are paying attention. First, I just want to do a little math. So when we talk about automobile engines and we talk about solar panels and stuff like that, the first question you should ask yourself is, how efficient is it? Do you want a gas guzzler car, or do you want a car that gets the most miles out of the same gallon of gasoline? And that really comes down to efficiency. So if we look at the current solar panels or current gasoline engines, the maximum efficiency is about 20%. That means it takes 20% of the available energy in gas and can turn that into kinetic energy of moving a car from one place to another. or 20% of the sun's energy and can convert that into electrical energy to store in batteries or use in electrical devices. So that's about the maximum that our technical knowledge is able to achieve. This might go up just a little bit, but it's somewhere around 20 to 25%. Now, let's figure out how efficient our body is from converting the energy of glucose into the energy of ATP. So remember, it's 7.3 kilocalories per mole of glucose in each ATP. So if we have 36 ATP for every mole of glucose, then we just multiply 7.3 by 36, and that gives us 268.8 calories. Remember, it's actually kilocalories per mole of glucose, which actually has 686 calories in it. So that means we're about 40% efficient at converting glucose to ATP, which is about twice as efficient as any current system we have of electrical conversion. And just to blow your mind a little more, remember this number is really closer to 13 to 15 in the cell. So really, we're about 80% efficient, or 4 times more efficient than any other mechanical device that's ever been created. That means that you're pretty amazing. Like, living things are pretty amazing at energy efficiency. All right. So just to recap, things that you need to know for the test. So what do you think would happen if complex 1 didn't work? How many ATPs would you make?
One second.
So let's draw it out. Okay, so we know that we have 10 NADHs, right? And of those 10 NADHs, if complex 1 worked, it would pass all those NADHs across there. We would pump 2 protons for each one. Every 2 protons equals 1 ATP. So we would expect to get 30 ATP out of the electron transport chain from NADH, right? 2, 4, 6. So 6 divided by 2 is 3. 10 times 3 is 30. Everybody follow that?
Mm-hmm.
All right. So if complex 1 was broken, would you be able to send the electrons through the chain from NADH?
No.
No. That means we don't get these 30 ATP. FADH2 doesn't need complex 1. It starts at 2, right? So we would still get those. There's 2 of these. We would still get 2 ATP for each one. So we'd get the 4 ATP from FADH2, and we would get the 2 ATP from the Krebs. So we would be able to make 6 ATP if this was broken. Make sense? Or you could just, if you knew it was 36 ATP, you could just subtract off the 30 that you knew you would lose from NADH, and that would give you the same answer. Make sense?
Yes.
Yeah.
All right. So these are the kind of things that you're going to have to start applying to the knowledge that you have to solving problems. This is done quite a bit because there are a lot of diseases that are thought to be caused by deficiencies in mitochondria, including Parkinson's and Alzheimer's disease. And so kind of knowing what happens, being able to predict what happens when you produce less ATP. And another thing is that we spent $10 trillion on anti-aging stuff, and as we get older, our mitochondria break down, and we make less ATP, which means less energy, right? And that's sort of what we equate to getting older. So if we could figure out these deficiencies in mitochondria or increase the number of mitochondria, you could be a multi-trillionaire because people would want your product. All right. Complex 2 is involved in the Krebs cycle and electron transport. Remember, we talked about that. I'll just back it up a notch, and you can just to remind you. So remember, complex 2 is part of the Krebs cycle, and it's also part of the electron transport chain. That's where FADH is. So what would happen if succinate was unavailable to the cell? So remember, complex 2's job is to turn succinate into fumarate and continue the Krebs cycle. So if complex 2 didn't have succinate, what would happen to the Krebs cycle?
It wouldn't work.
Yeah, it would shut down. So could we make ATP?
If complex 1 worked, right?
Well, yeah. So the Krebs cycle is out. That means you have to know what comes out of the Krebs cycle, remember? So it's ATP, we get 2 ATP, we get 6 NADHs and 2 FADH2s. So we're going to lose these ATPs. This is 3 ATP for each one, so that's 18 ATP that we can't get from Krebs. This is 4 ATP, remember, because each one of those is 2. And then this is 2 ATP from the Krebs cycle. So immediately, by losing the Krebs cycle, we're going to lose 24 ATP out of the 36. So that means we can still get 10. Where do we get those from? Or 12, sorry. Remember, we get 2 NADH from glycolysis. We get 2 NADH from turning pyruvate into acetyl-CoA. So that gives us, each one of these is worth 6 ATP. And so those are the 12. The answer is yes, it could make ATP. We would get 12. We would, from the NADHs that were made not in the Krebs cycle, but other places. What if complex 2 could convert succinate? So the Krebs cycle is running, but it can't oxidize. What does oxidized FADH2 mean?
Does it mean lose electrons?
Right. So it couldn't pull the electrons off FADH2, which means that how many ATP would we make? You can answer this pretty quickly by saying we make 36 ATP, right?
Yes.
And we know that we get 4 from FADH2. So we would make 32 ATP because we couldn't use FADH2. Does that make sense?
Wait, so we can get 32 because we could use FADH2? Or?
No, we can't.
Oh, okay.
We can use NADH. There's 10 of those, So we would get 30 ATP from that, and then 2 from the Krebs cycle. So 30 plus 2 is 32.
Cool.
So the reason I'm going through this is that I want you guys to know what goes in and what comes out, and you need to be able to keep tally of the energy that's being made. So you need to know how many ATP come from FADH2, how many ATP come from NADH, how many NADHs do we make, how many FADH2s do we make. Does that make sense?
Yes.
So you're just going to have to mathematically keep track of this stuff, because as scientists and stuff, like let's say someone comes in and you test their ATP and they're making 4 less ATP. What would your diagnosis be of them if they lost 4 ATP? What do you think is wrong with them?
It's not oxidizing.
Yeah, complex 2 is messed up. And so you wouldn't be able to tell that if you didn't know how many ATP you got from FADH2 and how this works. All right, so here's another question, potential test question. Cyanide and carbon monoxide, they're both toxins. They block the electron transport chain at complex 4. So just a reminder, we have complex 1, and then 2 is the peripheral. And then we have 3, and then we have 4. I'm not going to draw in the cytochromes or coenzyme Q, but there's coenzyme Q10 is there and cytochrome is there. So the electrons all flow, no matter where they come from, they go through complex 4, right? So what if complex 4 is broken? What happens to electron flow? What if I cut your vacuum cleaner cable? What would happen to the electron flow?
It would stop flowing.
Right. And so how many ATPs would we make then?
0.
It's not 0. How many do we get from electron transport?
36.
No. How many do we get from electron transport?
34.
34.
So how many ATPs could we make?
2.
That's right, exactly. So if you were to die from carbon monoxide or cyanide poisoning, the number of ATPs that you make is the same as holding your breath. So dying from this is essentially the same way that you would die if you held your breath. The difference between these two is cyanide is a covalent bond. Carbon monoxide is an ionic bond. So which one of these is reversible?
Carbon monoxide?
Yeah. So if someone came into your emergency room and they had cyanide poisoning, what would you tell them?
Sorry.
Yeah, that's it. There, you can't undo covalent bonds. But if they came in with carbon monoxide poisoning, what would you tell them?
Good news.
Yeah, you're going to be fine. We just got to get some oxygen to you to reverse the bonds that are going on inside of you. So one is super lethal, one isn't, but they bind at the exact same place. And it's because of the strength of the bond that it's either undoable or not. It's kind of interesting. All right, so what would happen if you leaked, the intermembrane leaked hydrogen ions? How much ATP would you make?
I don't know.
So remember, all those hydrogens make all the ATP in electron transport. So how many ATP do we make in electron transport?
34.
34.
Right. So if that thing was leaky, you couldn't make any ATP from electron transport. So you would only get 2 ATP from the Krebs cycle.
Okay.
Make sense?
Yeah.
All right. So you guys make sure you know the parts of the electron transport chain. Complex 1, complex 2, and their location. Coenzyme Q, complex 3, cytochrome C, complex 4. Make sure you know the path the electrons take for both NADH and FADH2. So remember, NADH goes through complex 1, skips 2, goes to coenzyme Q, then to 3, then to cytochrome C, then to 4, then to oxygen. FADH2 doesn't go through 1. It goes 2, CoQ, 3, Site C, 4, and on to oxygen. There's going to be some questions on the test like what I just asked you. So if there's something faulty, I might ask you on the test. You should be able to tell me what the outcome would be, and how many ATP are produced. It's the same thing that we just did on those exercises. Make sure you know the parts of the mitochondria. I'm going to ask you on the test. Where is the Krebs cycle? Where does that happen?
Is it the matrix?
Yeah, that's right. Where does glycolysis occur?
The cytosol?
Yes, exactly. Where is the electron transport chain? It's in the inner membrane. Where is F1 located from the ATP synthase? It's in the matrix. And where is F0 located? That's in the inner membrane. So those are the kind of questions that I will ask you, and that's why you need to know the parts of the mitochondria: the matrix, inner and outer membrane, and the intermembrane space, which is between the inner and outer membrane. What part of the electron transport chain is also part of the Krebs cycle?
The electric transport chain?
Which one?
Oh,
So remember, we have complex 1, 2, CoQ, 3, Site C, and 4. So which one of those is shared with the Krebs cycle? The answer is complex 2. Make sure you know that for the test. I will ask you that. What happens if succinate isn't available in the cell? How many ATPs will we make?
34. No, 34.
Well, remember, Krebs cycle shut down, but we still have the NADHs from glycolysis and from making acetyl-CoA. And we can make 6 from each of those, so that gives us 12. You should know Peter Mitchell introduced the chemiosmotic theory. Make sure you know that that ATP synthase is driven by protons, not electrons. The electrons' job is only to pump the protons, right? It's the protons that make it spin. Make sure you know the parts of the ATP synthase. So you need to know F0 and F1. Know where they're located. So remember, F0 is in the inner membrane. F1 is in the matrix. And remember, it's the protons, not the electrons. The protons drive this thing to make it spin to produce ATP. How many ATP do we make per NADH?
2. No way.
Try again.
Let's see.
6.
No, that's how many protons we pump, but not how many ATP.
Oh, yeah.
Remember, every 2 protons equal 1 ATP. So it's not 2, it's not 6,
4,
not 4.
8.
No,
12.
no.
Is it 9?
No. So here it is. Every NADH, we make how many?
3.
Yes. Congratulations. Yeah. So what about FADH2? How many do we make per FADH2?
1.
No. Remember, it goes through complex 3 and 4, pumps 2 protons for each one. Every 2 protons gives us 1 ATP. So how many protons? I mean, how many ATPs?
4.
No, that's how many protons,
6.
but we need
Oh,
2.
sorry.
We have 4 protons, but we need 2 protons to make 1 ATP. So 4 divided by 2 is?
2.
Yes. Awesome. Why does FADH2, sorry, there should be a 2 there. Why does FADH2 produce less ATPs than NADH?
Because it costs some.
No.
Oh,
So let's just recap this. So we have complex 1, 2, complex 3. I'm not going to draw coenzyme Q and stuff, and then this is complex 4. I'm sorry that 3 is awful. It's a 3. All right. So the electrons go through here from NADH. Remember, it goes through 1. So we pump 2 protons here, 2 protons here, and 2 protons here. Every one of these makes an ATP eventually. So we get 3 ATP, right? Remember, FADH2,
starts at complex.
right? So we only get 2
Okay.
ATP for FADH because it pumps 2 fewer protons. So FADH2 pumps 2 less protons. And that's why we get 1 less ATP from it.
Okay.
All right, we talked about this already, so you don't need to know that. This is just kind of my joke about how different things have different affinity for electricity. It's the, do you want to put a plastic fork or a metal fork into an electrical outlet?
Plastic.
Yeah, it's a little safer. Okay, so that is why we breathe oxygen and why we eat food. It's simply to make ATP so it can power, it can do all those magical things that we talked about, like move your muscles or decrease the Gibbs free energy so we can get reactions to become spontaneous. You know, we can bend molecules with it to make them weaker. To break, like it's funny because my 14-month-old, he likes to take soda cans or any kind of pull top cans, and he'll bend the tab back and forth until it breaks off. And I was like, oh yeah, that's a classic example of weakening a bond by bending it. And you know, then it pops off and then he thinks it's cool because it rattles inside the can. But that's how your body works, right? It's adding phosphates to bend those molecules back and forth to weaken them so that we can get a reaction to occur without changing temperature, which is, I think, kind of cool. Anyway, so you know, you're running on the treadmill or whatever, and you're sweating and you're increasing your oxygen rate and you're breathing heavier and you're breathing in more oxygen because you need oxygen at the end of the electron transport chain to accept those electrons, or your electron transport chain would shut down. And if you can't get enough oxygen, what happens to your electron transport chain? Like if there's no oxygen, there's nothing. It's at the end of the cord. Like if you took away the vacuum cleaner, It's not going to do anything. So you're not going to be able to make ATP through electron transport chain. And so if we took away all of your electron transport chain, you would only get those 2 ATP made in the Krebs cycle, right? But the Krebs cycle also requires the presence of oxygen to get it to flow. So once you hit that wall, once you run out of oxygen, you're breathing as heavy and as fast as you can. You want to run faster. That line's chasing you, but you just can't make any more ATP because your lungs are only so big. You only have so many red blood cells to carry oxygen. There's only so much of oxygen available. But you can still get a little bit more energy, just a hair, that maybe will save you from that lion or tiger or bear. And this is how it works. So in humans, this is what we do. We do something called lactic acid fermentation. And the reason let me just back up. So yeast and bacteria, they can do a fermentation which produces ethanol. We don't produce ethanol. So imagine what would happen if you're being chased by a lion and you ran out of oxygen, but you need a little more energy, maybe just to finish climbing that tree or whatever, and you're producing ethanol. What would happen? You guys know what happens when you drink ethanol?
You die.
Well, no, you get drunk, right? That's the same ethanol that's in like vodka or beer or wine. And so you probably would be like, oh, I don't care if the bear eats me or not, right? So animals don't do ethanol fermentation, whereas yeast, they're really not getting chased by bears and stuff. And so this is what happens. We have glucose. Remember I told you glycolysis happens no matter if oxygen is present or absent. We have to, we produce those 2 ATP. Remember, we put in 2, but we get 4, so we get a total of 2 ATP out. And we make pyruvate. So you guys have already seen glycolysis. You know that we make pyruvate. And if oxygen's present, that pyruvate would go into the mitochondria. And it would get converted into acetyl-CoA. And it would go to the Krebs cycle and then to the electron transport chain. But it doesn't do that because there's no oxygen. So instead, what pyruvate gets converted to is acetaldehyde, and that gets converted into ethanol, which is the same ethanol in beer and wine. And you also produce carbon dioxide. So if you ever made your own beer, you know that all you need is maltose, right, which is 2 glucoses linked together. You put it in, the yeast undergo fermentation. So you have to, it has to be in the absence of oxygen. So you have to put a contraption on it so that no oxygen can get in. And yeast will do this. So the byproduct of maltose is carbon dioxide, which is the bubbles in beer, and ethanol, which is what gives you the buzz in beer. And so that's how beer is made, by taking advantage of this alcohol fermentation. In humans, we don't do ethanol fermentation because we want to try to get away from that bear. So same thing, glucose goes through glycolysis. We produce 2 ATP, and then the pyruvate at the end of glycolysis gets converted into lactic acid. This is what causes your muscles to cramp. So if you're working really hard and your muscles start cramping up, you can blame your lack of oxygen and lactic acid for that. You also make the NADHs in glycolysis, but you use them to convert pyruvate into lactate. Same thing here. The yeast and bacteria make the NADH, but they need this energy to convert acetaldehyde into ethanol. So what I want you to take out of this is that fermentation occurs in the absence of oxygen. And then then that's showing you here. So here's the oxygen path, there's the non-oxygen path. Glycolysis occurs no matter what. And we end up with 2 ATP, and there's 2 different kinds. There's ethanol fermentation, which is done by yeast and bacteria, and that's how we make wine and beer. And then lactic acid fermentation is done by mostly animals. So bacteria do this, and this is in your yogurt and cheese. That's how we produce yogurt and cheese. Any questions about fermentation?
No, no, sir.
Okay. All right, so last slide, second to last slide. So we just focused on glucose, right? We focused on carbs, and glucose goes directly into glycolysis. And then we make that G3P, which I showed you, and then pyruvate's made. That's converted to acetyl-CoA, goes into the Krebs cycle, and then we get all those NADHs and stuff. But you guys know that you can use fats for energy. So the glycerol heads that we learned about in chapter 5, those get converted into glyceraldehyde 3-phosphate, which is bypassing the first part of glycolysis. The fatty acid tails of fats get converted to acetyl-CoA. Proteins get put into here in different pathways as well. And so it's skipping some of the steps in glycolysis by using proteins and fats. When this all comes down to nutrition, and this has a lot to do with those the ketosis and those, you know, keto diets and carb restricting diets or high fat diets or high protein diets or whatever. I'm just showing you that all these get broken down. They just get broken down in different steps. And in the end, ultimately, they all end up going to the electron transport chain, no matter where they go into the pathway that we learned about. Any questions about that? I don't want you to memorize this. I'm just telling you nothing that you don't already know, that you can use proteins and fats for energy, as well as lots of different kinds of sugars. We just use glucose, but you know, there's milk sugar and fruit sugar and all kinds of other sugars that you can use for energy too. We also can run this backwards. So if you need to produce fats, you know, if you sat around and ate carbohydrates, those would go through this pathway, and the excess ones would be produced, would be made into fats, and those would be stored in fat cells. If you need protein, you can convert these things into proteins if you need to. So you can not only break down these products, but you can also build them up, going through the same respiration process that we talked about. That's it. So that's chapter 9. So I'm going to end here and see if you guys have any questions about the test or whatever for the next few minutes. And then on Thursday, I'll do like a little mini review, and then we'll hit chapter. So we skip, no, chapter 10 is photosynthesis. So we'll do photosynthesis. We'll start photosynthesis on Thursday. This is all the material that will be on your next exam, which is, I believe, open tomorrow at midnight, and then closes on Monday at midnight. The 12th, yeah.
So the test is due at 12 at midnight, correct? By midnight.
Yes, yeah. So it has to be finished by then. So it's the standard same test. It'll be like 50 questions, multiple choice, or multiple guess, depending on how much you studied. And it usually takes about an hour for in-person students to finish the test, 50 minutes. You have longer than that. I give you, a couple hours, I think. But and then it's the same thing. You can bring a calculator, but it can't be programming or graphing. Scratch paper, show it to the camera, number it before and after the test. No notes, no earphones, no outside electronic devices except the calculator allowed. Yeah, and it covers chapters 6, 7, 8, and 9. So make sure you start it before midnight because you only have like a minute to finish it.
Would you say chapter 2 is the toughest test this semester?
It really depends on. I mean, this one is. There's a lot of material. Most of I'd say the majority of material comes out of chapter 9 because it's such a complex series of processes. But genetics is really mathematical and statistically oriented. And so that test is probably the hardest test, the final, because of the genetics and the molecular biology that's in that chapters 16 and 17. Up until now, it's been mostly memorization. Now it's starting to become applying what you learn.
Mm-hmm.
And so it tends to get a little harder for students. This is a hard class. It's a lot of material. It's it's like I said at the beginning, it's like you're learning a foreign language, chemistry, and biology all at the same time. And then mix in some math, and it makes it extra hard. Any other questions?
No, sir.
Okay, guys. So I'll just end it a little bit early, and then I'm going to jump off over to office hours around 2. So if you have any questions, you can hit me up there. Otherwise, I'll be back on Thursday. We'll do a review. So if you guys have any quiz questions you want to go over, or any questions about that, I'll spend the first 30 minutes reviewing, and then we'll continue with chapter 10. And then, like I said, you can take the test anytime you want up until the 12th. I would start it no later than 10 o'clock at night on the 12th. But anyway, you have some time to sort of digest this stuff and study it.
I have a question about the extra credit.
Sure.
That's due on Monday, correct?
Yeah, extra credits due the same time as the exams.
Okay, and do you have any specific requirements for it other than the question? I know it says it's a page, but do you want it paragraphed or anything like that?
Yeah, no, I don't. I just want you guys. The reason for the extra credit is so that you'll go out on your own and sort of learn about the relevance of all this stuff that you just learned. Why is it important for you to know this? And so I want you to kind of look up human diseases or diseases in general that are caused by defects in this so that you can see the relevancy of it. It's not really rigid assignment. I just want to see that you put forth some effort to see how this applies to the real world.
Okay.
And make sure you cite it so that I know that you're just not making up stuff.
Yeah, okay.
But I don't really grade it that harshly. I just want you to, throw out some facts and let me know that you've done some research on that.
Okay, with the citing, do you want it a professional cited page, or do you just want the websites attached?
You can just put the web links, it's fine.
Okay.
Question.
Thank you.
Any other questions?
No, not right now.
All right, guys. I'll see you on Thursday, then, you know, I'll have office hours tomorrow if you need anything, but we'll pick up lecture on Thursday.
Okay,
Okay,
thank
thank you,
you.
Professor.
Have a good one. Bye.
You too.